Well, hi, everyone, and welcome to Research America's discussion today. I'm Mike Coburn, Research America's Executive Vice President, and thanks so much for joining us today. And thank you and your organizations for your partnership with Research America. Now, if your organization is not a member, you can join us. My colleague Anna Platt would be more than happy to connect with you to discuss the value that comes with being an Alliance member. Her email is in the chat, and she hopes you'll be in touch. But more importantly here, today we're so delighted to have with us Dr. Rick Woychik, who is the director of the National Institute of Environmental Health Sciences at NIEHS. Now we've asked Dr. Woychik to help shed light on the critical intersection, or should I say intersections, between human health and the environment and what's on the horizon for the Institute. And as always, please type your questions into the Q&A box or chat, and we'll ask questions and have a Q&A session with Dr. Woychik later in the session. So Dr. Woychik, I'm going to turn it over to you.
Great. Well, thanks very much, Mike. And I just want to double check, and everyone hear me okay? Terrific. Okay. Well, thank you very much for this opportunity to be with all of you this afternoon. And in the limited time that I have, what I thought I would do is, I'm not going to talk about everything we do. I can't do that, but I've chosen a few projects that I think will illustrate some of the exciting new scientific directions that we'll be exploring over the course of the next several years. So let's go on to the next slide, please. So if you're not familiar with NIEHS, I'll take just a minute to discuss our mission and our vision. So we are the Environmental Institute of the NIH and the 27 institutes or centers of the NIH, and our mission is all about discovering how the environment affects people in order to promote healthier lives. And our vision is to provide global leadership for innovative research that improves public health by preventing disease and disability. Now, the key here is the word preventing, and this is integral to our vision because our strong focus is about identifying and understanding those elements of the environment and the environmental exposures that can either positively or negatively impact human health. So we're not about discovering cures for human disease, which is, as I think most of you know, is a focus of many of the other ICs at the NIH, but rather we're about preventing disease from happening in the first place. Next slide. So we have a strategic plan in place, and in the interest of time, I'm not going to go through all the details here. If you're interested in knowing the things that we support and what we do, take a look at our website. It's www.nihs.gov, NIEHS, and it will help you understand the three different themes of our strategic plan. So the next slide, please. So we're interested in studying the environment. And what this slide illustrates is that studying the environment is not so easy because we define the environment very broadly, and it includes a whole variety of different exposures, from chemicals found in products and building materials to pollutants in the air that we breathe, pesticides and agricultural chemicals, and cosmetics and sunscreens. So we also analyze the chemicals in our workplaces, as well as the influence of lifestyle factors such as nutrition, exercise, smoking, vaping, and psychosocial stress. So the bottom line is we're interested in a broad assortment of different environmental exposures and testing and evaluating how these can positively or negatively impact the biological networks that drive human biology. But next slide, well, click, please. So to make our task even more complicated and challenging, I think it's part of the reason why I, as director of NIEHS, have a genetics background. So we recognize that we have to account for differences in the way that each of us responds to an environmental exposure. So what we come to appreciate over the years is that different individuals with their own genetic and biological makeup respond to the environment in different ways. So what may be dangerous or what makes someone sick at a given dose for one individual may not apply to someone else. So Spencer, if you could click again. So what's emerging from many members of our community, the environmental health sciences community, is an exciting new concept. It's called precision environmental health, where we study the gene by environmental effects. So this gives us a better understanding of the genetic, the epigenetic, and the biological basis for why individuals have different responses to various environmental exposures. And so we feel that this program integrates very nicely with the precision medicine program that many of you may have heard about at the NIH. But it's not just about genetics, it's also integrating environmental exposures and, say, epigenetic changes in the genome that be influencing how our genes actually do their job. Next slide. So to set the stage for a discussion on genes by environment experiments, this is a slide that clearly shows the influence of genetics on differential response to a couple of environmental exposures. On the left is acetaminophen toxicity, and on the right is benzene clearance or benzene toxicity. So what you can see on the slide, and we use different inbred strains of mice for these experiments because we can control the genetic background. So as you look at different inbred strains, the toxicity response of acetaminophen or of benzene is vastly different. And that's because there are different genetic backgrounds. They're all furry rodents, but their genetic background is different. So the bottom line here, just in the interest of time, I'm not going to go through a lot of the details, but the genetic background can influence how an individual, whether it's mice or humans, respond to an environmental exposure. And the good news is that we have some very powerful tools now that we can bring to bear to better understand the molecular reasons for these differential responses. Let me go to the next slide. Now, a key to using a gene by environment framework is to get to the point where we can understand the genes and the variations within those genes that are responsible for differential responses to environmental exposure. So NIEHS partnered with NHGRI and several other ICOs across the NIH several years ago. And that was to launch what we call the Genes, Environment, and Health Initiative. This was a nice starting point to link genetic susceptibility to exposures and disease. This was a collaboration that we think was pretty successful, and it provided a lot of useful data, especially in developing, say, GWAS data around environmental exposures. But what was clear is that genetic traits associated with environmental exposures fall into this category that we refer to as complex traits, which means that there are many, potentially hundreds of different genes that are associated with an environmental exposure or that can dictate differential responses to an environmental exposure. So the key going forward is to identify the gene variants associated with these complex traits. And again, because it's not a single gene or a single gene variant, it's complicated. So next slide, please. And what we've learned is that ultimately identifying gene variants associated with complex traits like environmental exposures, responses to environmental exposures, it requires extremely large sample sizes, especially when you're studying human populations. So common variant association studies require tens of thousands of cases to have reasonable statistical power to exhaustively detect disease-associated loci. And those rare variant association studies in coding regions requires even larger sample sizes, potentially hundreds of thousands of cases. So therefore, I was encouraged by the leadership of Dr. Collins, the former NIH director, and Dr. Eric Lander, when they pulled together the international genetics community to launch the formation of what they refer to as the International Common Disease Alliance. This happened back in September of 2019. So this Common Disease Alliance, it's referred to as the ICDA, and it's really all about coalescing international support and collaboration around what this concept of the M to M to M challenge, which is maps to mechanisms to medicine. So in brief, we discussed how we need more comprehensive genetic maps of complex traits, and we need to understand the molecular mechanisms of common disease. And we need to develop the processes to use this information for the practice of medicine and for promoting public health. So the ICDA is about bringing together the international human genetics community under a shared vision to collaborate on developing new ideas and principles. So the ICDA will undertake foundational research projects, create comprehensive gene catalogs, will develop new technologies, and then in the end, we'll democratize these resources. There can be seamless sharing across different research programs across the globe. So in the end, we're hoping that this could ultimately we could end up with tens of millions of potentially very highly genetically diverse individuals across the globe And that would be an ideal place to begin to study those gene variants associated with differential exposures to differential responses to environmental exposures, though G by experiments. But next slide. So, but to do that, engaging with the ICDA community, which has been pretty much genetically focused, we, as the environmental health sciences community, have been working with them to integrate environmental exposures into the framework of the ICDA. So my colleagues and I at NIEHS feel that this is important because we come to appreciate that environmental exposures can give rise to many disease phenotypes that are part of the ICDA. Kind of the bottom line is those environmental exposures, lead, asbestos, air pollution, a whole variety of different things, as you can see on the slide. PFAS, those perfluorinated alkyl substances, Teflon, Scotchgard, these can make people sick. So if the ICDA is ultimately interested in essentially enabling precision medicine and correlating misbehaving gene functions with disease phenotypes, we feel that it's critically important that we integrate environmental exposures into the ICDA framework. Next slide, please. I mean, just as an example, if you look just at air pollution, it's clear that people get sick and they die from bad air that they breathe. In fact, according to the Lancet Commission report on pollution and health in 2017, air pollution is one of the most significant contributors to morbidity and mortality across the globe. It accounts for 16% of all deaths worldwide, which is 3 times more deaths from AIDS, tuberculosis, malaria, and 15 times more deaths than from all wars and other forms of violence. So as you can see on this slide, air pollution alone accounts for a wide assortment of phenotypes associated with respiratory, cardiovascular, neurological, immune, metabolic, and neonatal common diseases. And these are all part of the phenotype portfolio of the ICDA. Next slide. So as closer to home at NIEHS, we've been working with Josh Denny and others in the leadership group at the All of Us program at the NIH. That's the Precision Medicine million-person cohort. We've been working specifically to integrate air pollution and other types of environmental exposures into this precision medicine program. So at the first level of collaboration, we're examining how to integrate geospatial data. And this data is currently available from other agencies like USGS, CDC, and NOAA. So we're doing this because knowing the address of where someone lives and where they work, this could provide extremely useful information and data on environmental exposures like the particulate matter 2.5 from air pollution, excessive heat from climate change, and social vulnerabilities following public health emergencies like hurricanes and wildfires. But this is just a start, and we hope to be continuing to work with the All of Us program to add many other environmental exposures that can ultimately predispose to human disease phenotypes and contribute to the program. So next slide, please. So I also want to just briefly mention here that certainly we can look at the variation in genetics, but we're also realizing that our thinking about individual variation in the not so distant past was a little bit too simplistic. And being a geneticist, I'll be the first to admit this. We used to think that the only effective way that you can change the expression of a gene was to mutate the DNA, change the base sequence. But now I'm sure you all recognize, we come to realize that methylation of the DNA, or many different types of post-translational modifications of different histones. These can also influence how our genes are being expressed. And of course, this falls under the banner of epigenetics. Next slide. And a great example that I frequently use to illustrate the impact of environmentally mediated epigenetic regulation of gene expression is work from my own laboratory. We started this over 20 years ago. It's the analysis of this gene called agouti. So if you look at the slide, you would assume that if you're a geneticist, you would assume that each of these mice have different mutations since they are clearly phenotypically different. Some of the mice are actually, there's an 85-gram yellow pigmented mouse on the right compared to a 25-gram littermate. But remarkably, all of the mice on this slide are genetically identical. It's one of the advantages of using these inbred strains of mice. But they all carry a mutation in this gene called agouti. The mutation is specifically called viable yellow. We originally characterized this, again, over 20 years ago. And this mutation specifically directs the normal expression of agouti, the agouti signaling protein. It directs it to be expressed ubiquitously in every tissue of the animal, including the hypothalamus. And so what we ultimately determined is that this ectopic expression of the agouti protein, the signaling protein in the hypothalamus, effectively short circuits the satiety response. So the animals keep eating and become massively obese and develop type 2 diabetes. So most notably, the animals are phenotypically, those animals all the way to the right, for example, that have the normal coat color and that they're phenotypically normal. There's an epigenetic alteration in the mutation, the agouti mutation, that suppresses this dominant trait. And what I don't have time to show you is that this epigenetic effect is influenced by the types of environmental exposures that mothers are exposed to during pregnancy. So for example, feeding mothers high levels of chemicals that fall broadly into the category of, quote, methyl donors, it substantially increases the number of pups that are phenotypically normal that are born. So whether a gene is expressed or how it's expressed, it can be influenced by environmentally mediated epigenetic changes in the genome of the animal. So I also don't have time to show you, but there's a window of exposure, and it's critically important. This environmental effect happens when mothers are pregnant. So, and again, I don't have time to show you the data. I'd love to, since it's from my own laboratory. But agouti is not the only gene whose expression is influenced by this environmentally mediated epigenetic change. So, next slide. So this is just very briefly to illustrate that many years ago, the NIH launched the Epigenomics Roadmap program. We did this in collaboration with NHGRI. And so there's a website now where you can actually look at different tissues, different human tissues, and you can look over the genome and get a better understanding of some of the epigenetic marks associated with, this would be normal tissues in humans. So you can actually compare that then to cases of tissues from individuals that are suffering from specific diseases. So I'm looking at the clock and I'm just a little bit over halfway through. So let's go to the next slide, please. I just want to comment on, so environmental exposures are important, but given the importance of environmental exposures and disease phenotypes, the Environmental International Health Sciences community has launched a new project called the Exposome. So it's more than just the latest ohm, but the Exposome is a research framework for evaluating the totality of exposures, of environmental exposures, and it goes beyond studying one exposure at a time. So Exposomics experiment involved measuring a broad spectrum of the types of environmental exposures that are shown. And you can see that there are a whole variety of things that we're showing on the slide on the right-hand side here. And these range from diet and psychosocial mental stress to different chemical exposures. So as you can imagine, supporting the exposome will involve developing innovative new tools for measuring individual elements of the exposome, and we're not there yet. So part of the reason for my showing this is that Exposomics work is a major focus of the Institute, and we're working right now with the International Environmental Health Sciences community to essentially operationalize the exposome. So if I wanted to do an exposome experiment, if you listening on the webinar want to do an exposome experiment, we will have more definitions for you. And how do you actually do the experiment? And furthermore, where do you capture where do you share the data once you've captured that data? So let's go on to the next slide. So again, I think just in the interest of time, kind of the bottom line here is that we're working with the International Environmental Health Sciences community to figure out how do we operationalize the exposome so that we all know how to do an exposomics experiment. Let's go on to the next slide. Again, in the interest of time, I'll just tell you that we have a a program at NIEHS. It's called the PEGS program. And the PEGS refers to the Personalized Environment and Genes Study. So if you're interested in the details, I encourage you, go ahead and get on our website, and we would welcome you to actually volunteer and to sign yourself up for the study. Next slide. Let's go to the next slide. And I just wanted to comment that there are two key investigators at NIEHS, Dr. Jan Hall, who's our clinical director, and Dr. Allison Matzinger-Reif, who's the head, the chief of our biostatistics and computational biology branch, are the key points of contact for the PEGS program. So if you're interested and you want to know more of the details, these are the two individuals to contact, or send me an email and I'm happy to make a connection for you. Next slide. So the last thing I want to take a few minutes on, it's a very important new program here. I want to update you on recent progress on the efforts that we've been making on addressing the challenges associated with climate change and health. And I believe, as all of you have heard, the president has made it clear that addressing the issues related to climate change is a very high priority for the Biden administration. So he also recognizes that there are a number of very important health-related issues that need to be addressed with climate change. So therefore, he issued Executive Order 14008, which is about tackling the climate crisis at home and abroad. And there's actually encouraging language in the president's budget for FY23 to add $100 million to the budget for NIEHS. But we're at a point, we're not certain that we'll actually get the money yet, but it's encouraging that it's in the president's budget. But nevertheless, we're developing an NIH-wide strategy for climate change and health. And so we're doing this on an NIH-wide scale because our feeling is, and certainly my feeling as director of NIEHS, is this climate, the health aspects of climate change are much bigger than what any one IC can do on their own. So therefore, what I've done is I've reached out to several other IC directors, most notably Drs. Bianchi at NICHD, Dr. Gibbons at NHLBI, Dr. Glass at the Fogarty International Center, Dr. Gordon at NIMH, Dr. Perez-Gable at NIMHD, and Dr. Zank. And together we formed what we're calling the Executive Committee for Climate Change and Health. It's an NIH-wide program. We've re-energized a NIH-wide working group that's co-chaired by Aubrey Miller and Josh Rosenthal. Well, Aubrey is at NIEHS and Josh Rosenthal is at Fogarty. And Dr. Gwen Coleman at NIEHS is providing much needed central coordination to keep all the working parts moving in synchrony with each other. So if you're interested, well, let me go on to the next slide. So one of the most important things, in just the last few minutes that I have here, one of the most important things that the working group has done is to create a strategic, NIH-wide strategic framework for climate change and health. And they developed this with a lot of input from the extramural community. They issued an RFI last August. We had over 180 different substantive responses. So you take a look at the framework. There are 4 basic core elements, which include health effects research, health equity, training and capacity building, and intervention science. And this will all be done with a multidisciplinary transformative research focus. It'll be conducted with global cross-sector interagency and community partnerships. And the cogs on the outside of the diagram illustrate different types of science that will need to be done to support this strategic framework. So again, I just encourage you, take a look. And now I don't have the URL on the website, but if you're interested, and I encourage you to take a look at the website, it's nih.gov/climate and health. That's one word, climate and health. So take a look and stay tuned. So we're working, we're hoping that the $100 million that can help to fuel this NIH-wide initiative will come through in the next year's budget. And but even if not, I think there's a lot of support across the NIH to get things moving. So next slide. So again, we're doing so many things, and it's so many things that really fire me up. And I wish I had time to go over all of these, but I don't. So I'm going to bring this to closure here, but I just wanted to point out that, I talked about climate change and health, precision, my timer is going off. Talked about precision environmental health, exposomics, but we're doing a lot of work on environmental justice and health disparities, working on developing our IT infrastructure to support, computational toxicology and computational biology, as well as managing our data. There's a lot of work being done planning around mechanistic toxicology. So, I don't have time to discuss these other areas, but I want to make sure we have time for Q&A. So let me stop at this point. Next slide. Thank you all for this opportunity to get out in front of all of you, and I'm happy to take any and all questions that you may have. So Spencer, if you could stop sharing the screen, that would be great. Mike, back over to you.
Well, thank you for all those insights, Dr. Woychik. And let me ask my colleague Anna to join us on screen to share some questions for the audience. And as I listen to your conversation today, I wonder, looking at public health, in terms of climate and the environment, where does that rank in terms of priorities in public health?
Well, it ranks right up there. And what I try to illustrate is that, but I think it ranks, it's more than what one IC can do. So for the past 10 years, NIEHS has been if you just count up the number of grants that have climate change, an area of climate change focus, I think NIEHS was one of the leading institutes. But I'll tell you, others have really stepped to the plate. And I noticed there's a question that's coming in around where's the NCI in all of this? Well, let me assure you that the executive committee formed with 6 other IC directors and myself, but we can't have an executive committee of 27 IC directors. So it's really more of an administrative formality. Let me assure you that unfortunately Ned Sharpless is stepping down from his role, but Ned and I have been having some very, very in-depth discussions about collaborative partnerships between NIEHS and the NCI. So as it relates both to climate change, but also more substantively around a broader base of interest around, what are those environmental exposures that predispose to the development of cancer? So stay tuned on that. There's a lot of planning and discussions, and those wheels are turning in the background. So as things begin to emerge, maybe you can invite me back and I'll have a discussion. Maybe have Ned and I, or the new NCI director and I, co-chair a little panel discussion. All right. Does that answer your question, Mike?
Yes, it does. Thank you. And Anna, let me turn to you to see what questions have come in.
Sure, absolutely. And just as a reminder to folks, please feel free to put your questions in the chat or the Q&A box, and we will do our best to ask them. And one question that's come in, Dr. Woychik, can you define environmental justice and then maybe share what you see as the greatest challenge in that space?
Yeah, well, environmental justice, I'm really glad that you brought that up, and I'm sorry with the limited time that I have, I didn't go into that, but environmental justice has been part of the fabric of the work that we do at NIEHS. So environmental justice is really recognizing the health disparities and the types of exposures that happen to certain communities that are disadvantaged communities. So for example, we recently had an environmental justice webinar, and we invited people from our own backyard. And these are people who are living next to Superfund sites, or living next to some of these very large farming operations. Any large animal farming operation creates a little bit of doo-doo. So, there are some pretty significant environmental concerns there. But environmental justice is really about rolling up our sleeves and figuring out how do we deal with not only the obvious health disparities, but also some of the environmental triggers that may be responsible for those health disparities. So I don't know, there's not a lot of time to go in depth, but does that answer the question, or does that address the issue?
Yeah, absolutely. And if not, I'm sure the person will ask a follow-up if they'd like. So another question that's come in, is this person's asking, they say that anecdotally, it seems like more folks in the US, and this person has read in other countries, are confronting a growing prevalence of asthma and allergies. And is this person's asking, is that an accurate characterization? And if so, is the environment a major driver in that?
Well, that's a great question, and I will essentially ditto on a whole variety of other human health conditions. And as a geneticist, you might ask, well, if it's caused by genetics and genes exclusively, well, have people evolved that quickly that we have all these gene variants that are responsible for these? Or is there something else that may be kind of percolating in the background, or maybe in the foreground, that's responsible for this increase in the incidence of asthma, incidence of ALS, incidence of Alzheimer's, I mean, I could go on and on and on. And the bottom line is that we feel that the environment is playing a critically important role in causing the acceleration of many of these adverse health effects. So a personal passion of mine is developing the strategies to work more effectively with the other institutes and centers across the NIH. I alluded to the conversations I've been having with Ned Sharpless. Well, I've been having the same conversations with Walter Koroshetz, for example, at NDS. What are the neurological disorders? And I think that's been, and Gary Gibbons, as it relates to asthma at NHLBI. So if you add up all of the genetic variant contributions to asthma, to ALS, to various neurological diseases, maybe you can account for 20, 25% of the contributions to triggering the disease. So I think there's just an increasing awareness that the environment plays a very important role. And hopefully one of my take-home messages from this talk is the environment can make you sick. And we need to be integrating studying the environment into the way that we study the etiology of human disease. So I don't know, I went on a little bit longer there, but do you think that addresses the question?
Absolutely, thank you for that. Another question that's come in, this person's asking, compared to, let's say, a few decades ago, is it easier now to disentangle the impact of environment versus other variables on diseases like cancer, for instance?
Well, it's still complicated, and I think we're making progress. And so I mentioned the Exposome project, and I also mentioned that we're spending a lot of time to operationalize the exposome. So for many years, the exposome was a strategic framework. And I think the key thing is that we have to get beyond studying one exposure at a time. Because the reality is that I'm sitting in my office at home right now, I'm breathing in a bunch of flame retardants, okay? That's just the nature of the indoor air pollution. But if I go outside, I'm going to be breathing in I'm going to be breathing in ozone. I'm going to be breathing in some of the effects of, the Greyhound bus that just passed through the community. So if we want to understand the environment, we have to take this approach to understand all of the factors that are contributing to our biology. And that's why I also mentioned the concept of epigenetics. So we need to be sophisticated looking at all the different variables. But in the end, our health is going to change if our genes start misbehaving. And our genes can misbehave because of those epigenetic changes that occur in your genome. So that's what we're pretty excited about, beginning to characterize that. So to answer your question, I think we're getting to the point now, just like with the Genome Project, we were doing genetics. 20, 30 years ago, we would study part of one gene, because that's the only technology we had. And then we would extrapolate up to what does it look like for 23,000 different genes. So we now have the ability to sequence the entire genome. So we can start out with knowledge in the entire genome. We want to get to the point where we can effectively start an experiment to evaluate the environment by looking at the totality of exposures. So does that think that answers the question?
Absolutely, it's fascinating, thank you. Another question that's come in, this person's asking, do changes in the environment affect infectious disease patterns, and have those evolved over time from what you have seen?
That's another great question. So Something that I happen to be very interested in at the moment, and I suspect that many of you are also interested in, is why the heck does SARS-CoV-2 have such a vastly different cause such a vastly different clinical response? Some people who are infected with the virus die from the virus, and some people are totally asymptomatic. They don't even know that they're infected. And then there are some of those people who are infected with the virus, even though they may be asymptomatic during the infectious period, they then go on to develop what we call PASC. It's that post-acute sequelae for COVID-19, or something we call the long hauler effects. So what is responsible for all these differential effects? So people are sequencing the genomes and taking a look at genetic variants. But we know, for example, if you take just these perfluorinated alkyl substances, and these things are persistent in the environment. The Teflon that was manufactured decades ago, it's still around. So those carbon-fluorine bonds, they're going to be on the planet Earth forever. So, and we know our grantees are doing experiments, and they know that these suppress the immune response. And we know that there are a whole variety of other, there are endocrine disrupting chemicals that can be influencing immune response. So what we're trying to do now is to begin to explore how does differential environmental exposures influence infectious disease with SARS-CoV-2 and a variety of other infectious agents. So I use SARS-CoV-2 as an example because it's very prominently in the news right now. And we have some of our grantees have actually correlated the degree of air pollution within different communities and the severity, the clinical severity of COVID-19. So does that answer your question?
That does. That is really interesting. Thank you. I think we might have time.
So again, stay tuned on that one. And
Absolutely.
there's a lot of stuff that we're doing. And I really have to thank my colleagues throughout NIEHS. They have really risen to the challenge to address the issues around COVID-19. And it's not over yet, folks, fortunately.
That's right. That's right. Exactly. So we might have time for one or two more questions. But this person has asked, it seems like the International Environmental Phenotype framework you're creating could also be used to explore other factors influencing illness, like hereditary factors. Do you see that as a model or a tool that could be applied beyond environmental factors?
Well, again, it's, so I'll just kind of refer back to my talk. I covered a lot of stuff very quickly. But the International Common Disease Alliance, I mean, that's about, that primarily was focused initially on genetics. And what we're doing now is overlaying environmental phenotypes. And you have health-related phenotypes. There's going to be blood pressure. There's going to be all the standard things that you would collect in the clinic. So we're thinking that absolutely, this ICDA can be about a broad base of research approaches for studying essentially I call it the etiology of human disease, which is basically understanding the molecular mechanisms that underlie how your genes are misbehaving when you suffer from adverse health effects. And does that answer the question, or was there another component in that I missed?
No, that was it. I think that answered it. And Mike, I believe we're just coming up to time, so I'll turn it back to you.
All right, well, I'm going to end with one additional question, very quick question, but just a question about your institute and how you're encouraging young researchers in training and in the various stages of career development to get into the field of environmental health research. How are you building a crowd of enthusiastic researchers?
That's another great question. I'm glad you asked it because we recognize, and certainly as director of NIEHS, I'll take full responsibility. A big part of my job is to make sure that there is longevity associated with environmental health sciences. So take a look at theme 3 of our strategic plan. And so it's really about ensuring that the environmental health sciences will have a trained workforce, an engaged workforce. And so we do this in many different ways. We have a number of different training programs where we get out into colleges and universities. We have specific programs that will get out into HBCUs and MSIs because we feel that a diversified workforce is really a strong and a critically important workforce. So we got the educational programs, but we're also hoping that there are a lot of people who are doing basic biomedical research right now, and we could benefit from them in the environmental health sciences community. So we're actually getting out there, actually in forums like this. So that's part of my job, is get the word out. What is environmental health sciences? But we think, and I think, and a big driver for me is collaboration. Bringing people with different areas of expertise, bringing them together, locking arms, and then venturing hypotheses that neither one of them would have identified on their own. So developing these collaborative relationships, drawing in people from the outside, training. We have programs where we get out into the high schools, actually into the grade schools, K through 12, and getting people excited about better understanding the environment. We also had public forums where even if you're not a scientist, we packaged up the work that we do in a way that we can describe it to people. Last Saturday morning, I gave introductory remarks to a women's health conference. And this was all about better understanding what are some of the things in the environment that if you got to pay attention to, especially if you're pregnant. So I don't know, does that answer your question?
It does, thank you. And I wish you a lot of success in this area, and we'll contribute as we can as advocates.
Well, again, thank you, and feel free to call on us at any time. And again, really appreciate you and your colleagues for making this opportunity available to me to get out in front of your membership.
Well, we thank you very much for joining us today. It's been a fascinating conversation, and we look forward to more advances in the work that you're doing and your colleagues. And again, it's been a terrific conversation. Thank you.
Excellent. Very good. Okay.
I have one quick announcement for everybody else before you sign off, and thanks for being with us today, everyone. I hope you'll join us again next week, next Wednesday, the 20th at 1 p.m. Eastern for another special Alliance discussion. We're going to have Dr. Georges Benjamin, Executive Director of the American Public Health Association and a Research America board member, join us to speak about the current state of COVID-19, where we're at now, what the future holds, and what's up, in the next possible pandemic. So there's a link in the chat where you can register, and keep an eye on your email for more information on next week's Alliance discussion. So with that, have a great rest of the day, and goodbye for now.